See if anybody's here yet. It's just 11 o'clock, so we'll give you a couple minutes before I start. This is our live WebEx video review for Exam 3. This is covering chapters 6, 7, 8, and 9. Hi, Lorenzo. So we're just, let me turn up my volume here. So it's just 11 o'clock, so give people a few minutes or so to log in before we get started. Lorenzo, if you have any questions, just unmute yourself and jump in. So this is our lecture review for Exam 3, covering chapters 6, 7, 8, and 9. And so 6 sort of sets the tone. That's a chapter that gives us a lot of little pieces that contribute to cellular respiration and photosynthesis. So kind of the background information or the foundational information that we need to understand those processes. Then 7 goes into photosynthesis, making organic glucose, our energy compound, from inorganic molecules, carbon dioxide and water in plants, using solar energy. And then 8 looks at how organisms use that glucose with cellular respiration. And then that really wraps up our metabolism unit. And then we're going to move on to genetics and inheritance in the next unit, and DNA. But chapter 9 sort of sets the foundation for that. We're kind of coming full circle here with our first chapters from the pieces of a cell, the chemistry behind that, our schnapps, and building our biological macromolecules through how we get and use energy and nutrients and those resources, where the schnapps comes from, and then metabolism, what we do with those, and then back to that big picture of the cell and the cell cycle. And then we'll move on to replication through reproduction and inheritance in the next unit. So we'll go ahead and get started. Again, Lorenzo, just unmute yourself and jump in if you have any questions at any time. I am going to share my screen with the learning objectives for this unit, and we're just going to go through those. I'm really going to go through them fairly quickly, just stopping to emphasize things that I know trip students up the most, the errors that I see, most common mistakes that I see, that I've seen in the quizzes, or that usually trip people up. So chapter 6, our metabolism. This is looking at a bunch of little pieces. This chapter, as I say in the lecture video, can seem a little bit disconnected, like we jump from one thing to the other. But all of those pieces fall under the category of energy and how we convert to chemical conversions to get energy, to release energy, to use energy. They're sort of the background foundational pieces and vocabulary we need to understand what's going on in photosynthesis and cell respiration. So to start with just this kind of broad overview of energy, we did some chemistry, now we're going to look at some physics. But potential energy, I have the ability that I'm not using yet to be energy. So potential energy is energy that's not being used right now, but can be. I just have to release it. So good image is water behind a dam. There's all sorts of potential energy in there. None of it's being used until we release that water and it flows over the dam. The chemical bonds in molecules, those bonds, those electrons that are either drawing the molecules together or being shared, are a great source of potential energy. To break those bonds and it releases energy. That's chemical energy, or the energy that's stored in bonds of big molecules. And then that energy, when we're actually energy in motion, kinetic energy, is our usable form. So mechanical energy As an example, it's the energy used for motion. That water flowing past a rotor in the dam to generate electricity is an example of kinetic energy. And then electricity, electrical energy is another example. Our two laws of thermodynamics that are related to what we're looking at, to our class. First law of thermodynamics is that energy cannot be created or destroyed. There is a set amount of energy in any closed system. We can't create it, we can't destroy it, but it can change forms. So in our system, energy comes from the sun. The sun has a finite amount of energy. It releases it. We get it on Earth. We capture it in our plants. The plants use that energy. They take solar energy. They convert it to chemical energy in glucose through photosynthesis, and then they break down that glucose in cellular respiration and capture that energy for growth, for movement. Plants use phototaxis. They follow the sun. For reproduction, for making pollen and ova in the plants for reproduction. So they use that energy and convert it to different materials. Then along comes a primary consumer that is going to eat the energy, the chemical energy in that plant, stored in the plant biomass, and convert it to mechanical energy as it walks, to chemical energy as it stores products as food. So whatever it's using that energy for. Every time we convert from one form of energy to another, it's not a 100% efficient conversion. So all of that energy stored in a molecule of glucose, when I convert it to kinetic energy in my muscles, and I contract my muscles and relax them to walk, not all of it goes into that movement, that walking. Some of it's converted to heat energy. So we measure heat as temperature. That's a measure of energy. The problem with heat, though, is that it dissipates very quickly. So it disperses quickly, and it takes more energy to trap it and use it in a workable form than we can get back from it. So we say that energy as heat is lost to the system, because I would have to put more energy in to get that form of energy into work, to be able to convert it to work. So I've lost that. So every time we convert energy from one form to another, we lose it. If we're not destroying it, it's still energy. It just disperses so quickly that I can't use it. That's called entropy. So entropy is the tendency for systems to move toward disorder. In the second law of thermodynamics tells us every time we do convert energy from one form to another, we lose some of it to the system as heat. So why that's important in biology, why we go to physics, because that tells us we need to constantly have an input of energy, that's sunlight, that has to be captured and converted. And as I move up those trophic levels, I continuously lose some of that energy as heat. What that means, plants always need more sunlight. Animals need to eat because I don't have 100% efficiency as I convert that energy. So I'm always going to lose some. I always need to continuously resupply my energy by eating or heterotrophs. Hi, Jakai, I see you joined us. If you have any questions, just jump in, unmute yourself, and ask. So our third, in chapter 6, we move from those laws of thermodynamics and energy. We look at metabolism, which is the sum total of all the chemical reactions in a cell. Those reactions can be catabolic, where I take big molecules and I break them into smaller ones. When I break those big molecules, I break those bonds and release energy. They can be anabolic reactions, where I use energy to build big molecules from small ones. So when I'm building molecules, I have to put energy in. So anabolic. So lots of words that are similar, but used in different contexts or with specific meanings. So anabolic reactions are those where I build big molecules from small. Catabolic, I break down big molecules into smaller ones. And we also learn the words endergonic and exergonic. Endergonic are reactions that release energy. When I break chemical bonds, I release energy. So I release energy when I take a big molecule and I break it into smaller molecules. If I'm going to build a bigger molecule by putting small molecules together, I have to put energy in. Did I get those reversed? Exergonic, I'm releasing energy. Endergonic, I have to put energy in. And we refer to coupled reactions. Coupled reactions are if I need to put energy in to a chemical reaction to build big molecules from smaller ones, I need to get that energy from somewhere. Where do I get it? I get it from breaking down a big molecule and using that energy. So these are coupled reactions. So anabolism, catabolism. Anabolism is endergonic, I have to put energy in. Catabolism is exergonic, it releases energy. Those are coupled. You should be able to look at a chemical reaction and know what the reactants are and what the products are. You have to look at which way the arrow is pointing, because I can write a chemical reaction with the arrow pointing in either direction, just depending on which way the reaction goes. And what's on the pointy side of the arrow, what the arrow points to are the end products. What starts it are the reactants. And we're going to learn a synonym for reactants in this chapter when we look at enzymes, which is they can be the substrate. So make sure you know which ones are the reactants, which ones are the products, and always look at the arrow. Which way is the arrow going? Because it doesn't always have to go left to right. So whatever's being pointed at, those are the products. And we learned about ATP. This is the cellular currency. This is what the cell can use for energy. Adenosine triphosphate. So it is a nucleotide, single monomer of nucleic acid with adenine as its nitrogenous base, ribose as its 5-carbon sugar, and triphosphate, 3 phosphates. And it's that third phosphate. There's a lot of tension between the second and third phosphate. It really wants to get out of there, so it breaks off easily. And when it breaks that bond between the second and third phosphate, that's the energy that gets released that the cell can use to do work. So that would be a catabolic reaction. I break a bigger molecule, adenosine triphosphate, break off a phosphate, I end up with 2 smaller molecules, adenosine diphosphate and a phosphate that releases energy. Our bodies are pretty darn efficient. We reuse molecules whenever we can instead of having to continuously regenerate them. So we also use energy that we get from glucose to stick that phosphate back onto an ADP to make ATP again. It's coupled reactions. I take my glucose, I break it, I capture the energy from releasing those carbon-hydrogen bonds, and I stick a phosphate onto ADP to give me ATP. Then I can use ATP to do work in the cell, and I break that third phosphate off and I capture that energy for the cell to do work. As Professor Fink says in his video, the glucose is like my $20 bill when I go into the laundromat. Yes, I have energy, but the machines can't use it. The washing machines and dryers can't use it. First, I have to change it to quarters. One of the things I'm not thrilled with having the chemical reactions for cell respiration show ATP as a product, because it's not, it's a coupled reaction. Glucose breaking down to water and oxygen releases energy that's captured for ADP plus P to join to form ATP. Those are coupled reactions. They should be side by side or one above the other. I don't get ATP from my glucose, just like I don't get those quarters from my $20 bill. There's a conversion that takes place in the change machine. The $20 bill doesn't become quarters. It's an exchange. So that's kind of more appropriate for ATP being generated from ADP plus P. That ATP is not made from the components of glucose. It's made from the energy released by and other reactants. So it's a separate equation. Just had to get that aside, clear that up. All right. So when we're talking about our metabolism, the sum total of all the chemical reactions that take place in a cell, every one of those reactions would take place anyway, right? Chemical reactions just occur spontaneously as those molecules or atoms come close to each other and their external electrons are able to interact and form bonds or break bonds. We can't make a chemical reaction happen that wouldn't. So we look at the periodic table. Are those electrons in that outer shell able to interact? Is this a possible chemical reaction? Yes, it can happen. No, it can't force it to happen. Can't make something happen that won't. But the naturally occurring rate that these reactions would happen at spontaneously is typically too slow for what we need in metabolism to maintain life. So we need what we need when we need it in order to sustain life in the cells. And so in order to make sure we have the products that we need and that we get rid of the reactants that we don't need, waste, things like that, our chemical reactions have to happen at the right pace. And it's much faster than would happen spontaneously. So we use enzymes. These are chemical reaction catalysts, and that word just means they make it happen faster. So enzymes are a subclass of protein that catalyze chemical reactions. Every single chemical reaction in our cells is catalyzed, sped up with an enzyme. That means we have loads and loads, thousands of different enzymes, because every specific reaction has a specific enzyme, because each enzyme is exactly the right shape for the reactant in a chemical reaction to occur. So we have chemical reactions that are stepwise in metabolic pathways. That's to control the release of energy or how much energy it takes. And so each step gets its own enzyme, and each enzyme has the exact specific shape to interact with its reactants, or substrate is what we call those reactants when there's an enzyme involved. So metabolic pathways are all the series of chemical reactions to get from the beginning to the end. Using cell respiration as an example, we have glucose plus oxygen as our reactants, ending in carbon dioxide and water. It's not just a boom, one step and done reaction. There are about 72 little arrows incorporated in that one arrow when we draw that reaction. Lots and lots and lots of middle steps going on. So those are our metabolic pathways, and each conversion of one substrate to a product, that product becomes the next substrate, which is converted and becomes the product, which is the next substrate, and each one of those has an enzyme. The induced fit model. So the way an enzyme works to increase the rate of reaction is it reduces the amount of energy we need to put in. Even reactions that are exergonic, that release energy, require a kickstart with a little bit of energy pushed in to get them going. They just release more energy than they require to get started. By reducing that activation energy needed, we allow these reactions to happen up to 1,000,000 times faster than they would anyway. So the way this lock and key model of enzymes tells us that the active site of an enzyme is the exact right shape for only one specific reactant or substrate. But a more precise model would be the induced fit model. If we induce something, we make it happen. And what that means is in our enzyme reactions, in our enzyme catalyzed reactions, when our substrate fits into the active site, that lock and key, it induces or causes a slight shape shift. The molecule, the enzyme, changes configuration just a little bit to kind of make that reaction happen. It makes it happen a little quicker. So the induced fit is when the substrate fits into the active site, it induces or starts a change to the enzyme that makes the reaction happen. And then the enzyme releases the product. The enzyme is now free to go on and act again. All right, I have this picture up here specifically because lots of people get this wrong because they don't look at the arrows. They read top to bottom. All right, so we got to follow the arrows. The arrows start at what goes in, and what they point to is what comes out. Our enzymes can be involved in either degradation reactions, where I take a large molecule and I break it down into smaller molecules, or they can be involved in synthesis reactions, where I take smaller molecules and I put them together to become a larger product. Right? Synthesis, this would require energy in. This would be endergonic, or it would be an anabolic metabolic reaction. But with the induced fit model, with our, oh, I'm not sharing the screen. I thought I was sharing the screen. All right, got it now? Sorry, thought it was up there. All right, so here's my induced fit model. Follow these arrows here. So this would be a synthesis when I've got enzymes involved and my enzyme is building a big molecule from smaller ones. Call it a synthesis reaction. Right? Synthesis reactions require energy, so they're endergonic. They're building big molecules from smaller ones, so they're anabolic. But those terms are kind of broad and general. If I'm looking at an enzyme catalyzed reaction, I would say this is a synthesis reaction, and this is a degradation reaction. I start with a bigger molecule, and I break it down into a smaller one. So I'm showing this image. Thank you for pointing out that it wasn't showing. To show, just like when I'm looking at a chemical reaction, I need to follow the arrow to see what the products and reactants are. When I'm looking at an image, I need to do the same thing. Most common mistake people make if they're looking at this image and I say, what are the reactants? They try to look at this top down and they say, oh, these two molecules here are the reactants. No, that's what the arrow points to. Those are the products. So these are my reactants, or the substrate that the enzyme will act on. Notice the enzyme active site is the exact shape of the substrate so that they can land here, that lock and key model, and form the enzyme substrate complex. So they come in, they latch on, I have my enzyme substrate complex. I get that slight tweak in the configuration of the enzyme to make the reaction happen. And I've broken my big molecule into two smaller molecules and I release these. That arrow is releasing the product. The enzyme stays the same, goes back to its original shape and is now ready to go find some more substrate and act on that. So the enzyme helps the reaction along, but it's not part of the reaction. It's not one of the products. It is then released, right? And it can go on and do more work. Here we're showing a synthesis reaction. This is the tail end of my arrow. This is where I start with two smaller molecules. They find, or the enzyme finds them. They're the exact shape for the active site on this enzyme. So they bind. Slight configuration makes those two small molecules into one big molecule, which is then released as product. And the enzyme's off on its own, back in its original shape, ready to conduct more synthesis reactions. So make sure when you see these images, I could draw this the other way, make the arrows go the other way. It would reverse what my product is, what my substrate is. So look at the arrows, right? Don't read top to bottom. Don't assume it's going to look the same. Look at the arrows for which way the reaction is running. And remember, enzymes are not changed by the reaction, by participating in the reaction. They are not part of the products. They are not part of the reactants. They are the enzyme. They're separate and apart, right? The analogy I use is in my lecture video is a matchmaker, a yenta, right? She's not part of the couple. Right? She brings the couple together. They go off and get married. She is not staying in that marriage. She's going off to fix up some more couples. Right? So not part of the couple, not part of the marriage, off doing her own thing. She just helps it to happen. Whoops. Let's see. Let me go back to my mouse and sent it flying. There we go. All right. So those are enzymes. That's the most common, one of the more common errors with this, with enzymes. So enzyme activity, look closely at any figures for that. Your lab, I think everybody did well in this lab. It helps you kind of figure these things out. Use the vocabulary. You will not get credit. So this doesn't apply to either of you who are here live on this, but to a number of people. Your book and your objectives are the source of information for this class. They are the accurate and correct source of information. So for those of you who Google what happens to proteins at high temperature, you're probably not going to get points unless you come up with the word that we are talking about, denaturing. That refers to a protein losing its specific three-dimensional shape. In enzymes, that means the active site is no longer the correct shape to bind with the substrate. So denaturing, high temperatures, heat, causes the hydrogen bonds to break that are holding that polypeptide in its correct three-dimensional shape. It's tertiary or quaternary if it's a larger multiple subunit protein. When it loses its shape, that is called denaturing. Your lab, you looked at factors that affect enzymatic speed. Temperature, as I increase temperature, molecules move faster, they're more likely to come into contact with each other. That is going to increase the rate of chemical reactions, including enzymatic reactions. As temperature increases, my rate increases until the temperature is so high that it begins to denature, to break those hydrogen bonds holding the 3D shape in place. The protein, those enzymes denature, they lose their shape, they can no longer do their job, and the reaction stops. pH, either high pH, so lots of hydrogen ions, or sorry, low pH, lots of hydrogen ions, or high pH, alkalinity, lots of hydroxide ions. Either one of those ions can interfere with the hydrogen bonding that's maintaining the 3D shape. So pH outside of the optimal range. So the optimal pH is where the enzyme has its form and is working best, so works at its fastest speed. As I add hydrogen ions and lower my pH, or hydroxide ions and increase my pH, either one of those will start to denature the protein, and the rate of reaction will decrease until the enzyme is completely denatured, has lost its shape, and can no longer do its job. The other factors were concentration. If I increase enzyme concentration, more enzymes mean more collisions with substrate, mean faster reaction. Or if I increase my substrate, increased collisions with enzymes, and so I increase my rate. For both of those, I can only increase to a certain level. I can only increase until every one of the enzyme active sites is filled with substrate. So at higher and higher substrate concentration, at some point my rate of reaction is going to level off. At higher enzyme concentration, the reaction is going to run to completion. Every single one of those substrate molecules will be converted to product much more quickly. So it'll be a faster reaction and it'll end more quickly. So those factors increase the rate of enzyme reaction. The presence of cofactors and coenzymes. These are molecules that bind or interact with the enzyme in a way that allows it to have its exact right configuration and work faster. A lot of vitamins and minerals are cofactors or coenzymes. They have to be present for that enzyme to work most efficiently, to work best. So we need those. Enzyme inhibition means stopping my enzyme from working. And a lot of times this enzyme inhibition, this is how our bodies know, okay, we can stop making this enzyme, stop this chemical reaction. And we have two types of inhibition. Non-competitive inhibition. Just think non-competitive, we're not competing. So the enzyme's active site is what we're competing over. So let's look at competitive inhibition first. Competitive inhibition means I've got some other molecule that's similar enough in shape to the substrate that it can actually land in that active site and block it. And that will prevent the enzyme from doing its job. It can't convert that molecule because it's not the right molecule. It's not the exact right shape, but close enough that it'll block the active site so that the substrate can't land in the active site. It competes with the substrate. So competitive inhibition means I prevent the enzyme from working by blocking the active site right there at the active site. Non-competitive inhibition means I have a molecule that's going to prevent the enzyme from working, but it's not going to directly compete with the substrate for the active site. And so the substrate can still land in the active site. There's nothing blocking it, but the inhibitor lands at some other place on the protein away from the active site, and it prevents that induced fit from making the reaction happen. Allosteric, we're going to see this word again later. Allosteric means at a distance or far away. So the inhibiting product or the inhibiting substance is going to bind with the enzyme at an allosteric site, somewhere far away from the active site or not at the active site. It's not competing for that space, right? But it's preventing the enzyme from actually completing that reaction. All right, questions on chapter 6? All right. Chapter 7. 7 and 8 are the toughies. I know there's a lot of detail in here. I can see you probably need to watch the videos and break this down, the best approach. If you're still struggling with this, one section at a time. Forget the whole chapter. Forget photosynthesis as a whole. Learn one section. When you know that one section, then move on. Don't think of it as an entire chapter because this is a lot. Same thing with cell respiration. If you learn each of the steps, it's a little bit easier. So photosynthesis, autotrophs, heterotrophs, pretty easy, straightforward. We've already looked at that. Photosynthesis, I'm going to capture sunlight energy to turn two inorganic molecules, carbon dioxide and water, into glucose. Autotrophs, self-feeders. Heterotrophs feed on others. Both of these have mitochondria and both of these undergo cellular respiration, but only my autotrophs use photosynthesis. So you should know the structures in the leaf, what they're used for. The stomata. So our stoma is mouth, that's the opening or the pore. We're going to get gas exchange, carbon dioxide. That's where it's going to come from. So stomata actually means like little mouth, but it's the pore. So that's how I'm going to get my carbon dioxide in, my oxygen out. And then in the chloroplast, they make stomata and stroma, similar words, just to mess with biology students, I think. But our chloroplast, remember, it's a double membraned organelle. It's got two membranes because it was originally a bacteria, so it had a membrane around it because bacteria is a cell that was engulfed through endocytosis by a larger prokaryote. So with that engulfing, it ended up with a second membrane outside of the first one. And inside that membrane, I have an aqueous solution, the equivalent of cytoplasm, but when it's inside the chloroplast, I'm going to call it stroma. And then that inner membrane, which has folded and folded back onto itself, so it actually looks like a stack of pancakes, many stacks of pancakes. That inner membrane is called the thylakoid membrane. The grana are each of the individual pancakes, and the thylakoid is the whole inner membrane. And chlorophyll is a pigment that captures sunlight. Know the overall process of photosynthesis by interpreting its chemical equation. So its chemical equation, carbon dioxide, 6 carbon dioxides plus 6 waters. Those are my reactants and will be converted into a glucose molecule plus 6 oxygen molecules. Those oxygen molecules are released. They're the waste product. I need energy to drive this reaction because I am building a big molecule, glucose, C6H12O6, from those 2 smaller molecules, carbon dioxide, CO2, and water, H2O. So I'm going to use sunlight energy, and I am going to convert that sunlight energy to chemical energy, to the potential energy that's in the bonds between the carbons and hydrogens in my glucose molecule. I am going to have to do this in 2 steps. So the 2 steps that I can divide my photosynthesis into are the light reactions and the Calvin cycle reactions. I used to refer to the Calvin cycle reactions as the dark reactions, just as a opposite to light reactions. But the Calvin cycle reactions can happen in light or dark. They don't care. So we can also refer to these as the light-dependent reactions because the light reactions can only take place when there is light present. And the light-independent reactions, the Calvin cycle reactions don't care if there's light there or not. They occur independently of solar energy being present. If I look at my total equation, H2O plus CO2 goes to glucose plus oxygen. I can divide those reactants based on where they go. The light reactions only use the water. They convert water into oxygen by stealing away those hydrogens. And then those hydrogens are carried to the electron transport chain. The electron transport chain generates ATP because I'm building a big molecule, so I need to put energy in, and the only energy my cell can use is ATP. So I take my light and my water, I split that water into hydrogen, I take my hydrogens to the electron transport chain, and I get ATP for my cell. And I release that oxygen as the waste product. So the H2O, the 6H2O going in and the O2 coming out, those are my light reactions. And I just take those hydrogens over to the electron transport chain to generate some ATPs that are then going to go to my Calvin cycle. Okay. So I've taken care of my water, the Calvin cycle. Now I'm going to use my carbon dioxide. And to get from carbon dioxide to glucose, from CO2 to C6H12O6, I need to put those hydrogens on there. So my NADPs are my electron taxis in photosynthesis. They carried those hydrogens over to the electron transport chain, generated some ATPs, and now those hydrogens are left looking for a place to land. They are going to go land on that carbon dioxide to make it glucose. So I can break my light reactions into H2O becomes oxygen, and those hydrogens from it are going to come into the Calvin cycle. My carbon dioxide that goes in is going to come into the Calvin cycle. That hydrogen is going to be tacked onto it for it to become glucose. So two separate steps in this. The light reactions give me energy and my hydrogens. The Calvin cycle reactions, now this is my sugar factory. Here I am going to put my hydrogens onto my carbon dioxide. In a gain of electrons, I'm going to reduce my carbon dioxide to glucose. Okay, the photosynthetic pigments, these are the molecules that let us capture that solar energy and use that energy for to convert to electric or to chemical energy. The photosystem is the complex of enzymes and photosynthetic pigments, chlorophyll A, chlorophyll B, carotenoids. Those are all different pigments. Chlorophyll A is the most active, but all of these can capture solar energy. So that photosystem is a combination of proteins, of enzymes in the membrane, in the thylakoid membrane, and the pigments that capture that energy that are used for photosynthesis to occur, for the light reactions to occur. The non-cyclic electron pathway, this is where I'm going to use that light energy to split my water. I'm going to use my electron taxi, my NADP, to grab a hydrogen and carry it to the electron transport chain on the membrane, on the thylakoid membranes. Just like in cellular respiration, I'm going to take that hydrogen, pull the electron off of it, bounce it down a series of membrane proteins, and use that to drive a concentration gradient with my hydrogen ions on one side. And then I'm going to release the electrons on the other side, and I'm going to let those hydrogens flow through this enzyme, ATP synthase. And as it flows, as those hydrogen ions flow through to reunite with their electrons, I'm going to generate ATP. Where's my ATP come from? From adenosine diphosphate plus phosphates. Where did they come from? They were broken apart from ATPs. Where did the building blocks come from? Food. So those are things that are present in the cytoplasm of cells, or in the cytoplasm or the stroma of my chloroplasts. So those parts are just kind of recycled continuously in there. So from nutrients, I get those from nutrients. We call that chemiosmosis, because as my hydrogen flows through, I'm using a concentration gradient for those hydrogens to flow through. So it's a chemical concentration osmosis. We're flowing through the membrane. So similar to osmosis in that we're going down a concentration gradient, but we are chemically making ATP through this process. So my end products from the light reaction, this non-cyclic pathway, I start with water, I split it, my oxygen is released as a waste product. The hydrogen, I'm going to separate the electron from the proton, bounce it down the electron transport chain to generate ATP. And then I'm going to reunite my hydrogenal ion with the electron. I have hydrogen now that is going to go into the Calvin cycle. That ATP that's generated in the electron transport chain is going to be used in the Calvin cycle to build glucose, a big molecule from smaller molecules from carbon dioxide. So the Calvin cycle has 3 steps. So carbon dioxide fixation, the first thing I have to do is get my carbon dioxide into a form where it is able to proceed to the next step of the reaction. So carbon dioxide fixation is I'm just altering my chemical composition of carbon dioxide. The Then the next part is reduction. I'm going to reduce my carbon dioxide by adding a hydrogen to it. Right? Reduction is the gain of electron in these processes. When I see the word electron, I'm looking for a hydrogen. Well, I just brought that hydrogen over on my NADPH. That's going to be oxidized by losing the electron. It's going to give up its hydrogen. It's going to give it up to the carbon dioxide. And when it gives it up to the carbon dioxide, that has been reduced. So carbon reduction is giving that hydrogen to the carbon dioxide. And then in the final step, I have to regenerate ρBP. So this is a cycle. So whatever I end up with, right, is going to start the process all over again. So my hydrogen comes in on my NADPH, my carbon dioxide comes in. When my carbon dioxide comes in to get fixed, the way it gets fixed is by uniting with ρBP. It unites with ρBP. It has now been fixed so that it's able to be reduced. So I reduce my carbon dioxide by giving it the hydrogen. And in that process, I get this molecule. Once I've reduced it, I get G3P. This is a precursor molecule to all sorts of carbohydrates. So I can put this together to make glucose. I can put it together to make fructose. I can put it together to make all kinds of carbohydrates. It's kind of like a monomer of a monomer. How's that? And this is what my product is. This will become glucose if I put two of them together. If I'm going to use it for energy in the mitochondria, I'll put these together to get glucose, or I can put it together into different ways to get other things that the plant needs. And by breaking off this G3P, I regenerate, I turn my final molecule in this cycle is back to ρBP. What I've finished with is now what I start with going into the next cycle. So we just keep going in a circle. So G3P, this is this molecule, we can say this is where glucose comes in. Right? G3P is a building block that I can put together one way to make glucose. I can put it, if I'm going to use this molecule right now, the plant needs some energy in the form of ATP. It's going to put this together as glucose. It'll go to the mitochondria, be broken down to generate ATP for the plant to use for metabolism. If the plant has plenty of energy, it might put this together as fructose to be stored in fruits on the plant. It might be put together as starch to be stored in the root of plants. It might be put together as cellulose to build new cell walls. So this is this nice subset of my monomer of glucose. This is a submonosaccharide, I guess you would call it. It's my building block that I can build all sorts of different things from, because the cell doesn't always need glucose. It could need other forms of carbohydrates. So in this process, this gives me the building block to make glucose or whatever other carbohydrate I need. You should take a look at the information on page 122 about photosynthesis, the impacts of tropical rainforest destruction and climate change. Clearly tropical rainforests, lots of photosynthesis. This is really where all the energy, lots and lots of energy is captured through photosynthesis. And since carbon dioxide goes into photosynthesis, this removes lots and lots of carbon dioxide from the atmosphere. If I chop down my rainforests, not going to remove nearly as much carbon dioxide. So if I'm adding more than I'm able to remove, we're going to continuously increase how much carbon dioxide is present. But a lot of times the way we destroy the rainforest is by burning it down. So maybe chop it down and then burn the wood that's left. So not only am I removing a carbon dioxide sink by removing rainforest, but I'm also increasing how much carbon dioxide is going into the atmosphere. So I get a double dosed increase. This can kind of snowball in that I'm removing a carbon dioxide sink and I am adding a carbon dioxide source, all in the same activity. G3P, we just talked about that a little bit, the different products that can be made for it, why it's such a good versatile molecule to be created through photosynthesis, because it can go and make glucose, as it shows in my photosynthesis reaction, or the plant can make other things from it. So look at that picture of the tree, tells you all the things that we can make from it. And then we've got alternative pathways, C4 and CAM. Then CAM, cactus, pineapple, tropical plants. C4, kind of dry, arid things. Corn is the classic example. And what these do, these are environments that are very hot and dry. And so if the stomata stays open on the plant, it would dehydrate the plant very quickly if it's open during daylight hours. And so the way plants that live in these very demanding environments, moisture demanding environments, either very, very hot, even though they're humid, or very, very dry. The way they deal with this is they separate out the light reactions and the light independent. So my light dependent reactions are my light reactions. My light independent reactions, my Calvin cycle, they separate them out. So C4 plants separate those two processes out by location of where they take place in the leaf. And CAM, my cactus plants, these separate out those two processes based on whether they take place in the day or the night. So the light reactions will go on during the day, but the stomata will remain closed so that the plant doesn't dry out, and then they will go through their Calvin cycle reactions at night so they can open the stomata and release the gas then. So that lets them deal with that stress without having to keep the stomata open all the time, or with just limiting where that gas exchange will take place so that they don't dry out. So those are my C4 and my CAM plants. Questions on photosynthesis? All right. Cellular respiration. I think this helps a little bit going and getting a double dose of that electron transport chain. The electron transport chain in photosynthesis and in cellular respiration work the exact same way. I just have fewer hydrogens going in for photosynthesis. It's just one from that hydrogen, or two from the water breaking down, from that H2O being separated out to oxygen and hydrogen. So I only get a few ATPs in the electron transport chain in photosynthesis, but I only need a few to go into that cycle to build my glucose. That's all I'm using this. I'm going to go build my glucose. In cell respiration, I want more ATPs because I'm using that for everything that the cell does. Right? In photosynthesis, I'm only using the ATPs to build glucose. In cell respiration, I'm using those ATPs for all of metabolism. So more hydrogens end up at the electron transport chain, so I generate a lot more ATPs. So cellular respiration, reverse equation, glucose, C6H12O6, and 6 oxygens. Go through a whole series of steps, and the end products are 6 H2Os, 6 waters, and 6 carbon dioxides. It's really what we exhale, those waste products. We divide this, well, I did 4 phases of cellular respiration, including anaerobic and aerobic. Okay, so we divide the process into 4 phases. Each of those phases, if you think glycolysis, has about 20 individual chemical reactions, each one with its own enzyme. So lots and lots of steps. And we do this in a stepwise fashion because if we broke all the carbon-hydrogen bonds in glucose all at once, it would be like a mini nuclear explosion. We want to make it a controlled explosion, releasing just a little bit of energy at a time, right? So that's why we do this in a stepwise fashion, but we just break it into 4 main steps. So Glycolysis is our first step, which is anaerobic. And then we have the preparatory reaction, the Krebs cycle, and the electron transport chain. These are considered aerobic because they can't take place unless oxygen is present. But we only use that oxygen at the very end of the process. But they do require it to be present just so we can get into the mitochondria to do those steps. All right, so our mitochondria, similar to the chloroplast, double membrane. We have an inner membrane and an outer membrane. That outer membrane came from engulfing our original tiny prokaryote. So we have the intermembrane space, is the space in between the two membranes. The inner membrane itself is that convoluted, floated, folded up membrane called the cristae. And then the matrix is the equivalent of cytoplasm. It's aqueous solution inside the mitochondria that contains all the dissolved substances. It's aqueous solution, all the nutrients, all the enzymes, all the things that are involved in this process. So glycolysis is my first step. The best thing to do to learn these is really look at those, the figures in your book that show what goes in, what comes out of each step. And you should be able to kind of do a flow chart of what's going in, what's coming out, where it's going. In glycolysis, this takes place in the cytoplasm. It is anaerobic, don't need any oxygen. And my glucose goes in. In this process, anytime I'm going to have a chemical reaction, I need to kickstart it with a little bit of energy. So I need to kickstart this by putting in 2 ATPs. Some of the substances floating around in that in the cytoplasm are my coenzymes, NAD, and NAD is my hydrogen taxicab. So I've got 2 hydrogen taxicabs floating around in there. And then because I've done work, because my cell is constantly doing work, I'm always breaking ATP into ADP plus P. I've got those ADPs plus Ps floating around. Glycolysis is going to go through a series of steps, and it's going to convert that glucose into 2 pyruvate molecules. So it basically takes my glucose and breaks it in half. And when it does that, 2 of the hydrogens from glucose are going to break off and go into their hydrogen taxis. It's going to turn my NAD into NADH. So I end up with 2 NADHs, 2 pyruvates, and I get 4 ATPs from this process. Those ATPs are generated when I break the carbon-hydrogen bonds. So I have 4 ATPs, sorry. 2 of those are going to go back and start the process all over again. So I have a net gain of 2 ATPs. Right, so every time 4 come out, 2 go back to start the process again. So net gain, I get 2 ATPs. What happens to those products? The 2 ATPs go off somewhere in the cell and do work. They get broken down to ADP plus P to get put back together. So 2 ATPs to do work. My taxis, where my NADHs go, those taxis, if oxygen is present, are going to move into the mitochondria. Their job is to carry hydrogens, drop off the hydrogens, come back for more pairs. So hydrogens are their pairs. So those NADs become NADH and carry 2 hydrogens into the mitochondria and drop them off at their destination, the electron transport chain. And then those NADs can come back to pick up more pairs. The pyruvate goes into the next step of the process. Here, the book kind of does an aside here and tells us what happens when oxygen's not present. I'm not going to do that. I'm going to come back to that, right? My pyruvates in cellular respiration, when oxygen is present, are now going to be inputs into the next step, the preparatory reaction. Just like it says, the preparatory reaction is preparing my molecule. So in this stage, my preparatory reaction, my pyruvates come in, they enter into the mitochondria because oxygen's present. My pyruvates aren't ready to go into the Krebs cycle. they need to be prepared for the Krebs cycle. And the way I'm going to prepare them is I'm going to combine them with coenzyme A. Coenzyme A is something that was obtained through nutrition, through eating, and it's now dissolved and it's in the mitochondrial cytoplasm, the matrix. It's in there and it's ready, and it's going to be tacked on, and that is going to convert my pyruvate into acetyl-CoA. To become acetyl-CoA, I also need to lop off a carbon and some oxygens. So I'm going to get carbon dioxide, 2 carbon dioxide molecules from this, plus acetyl-CoA. The carbon dioxide is exhaled as waste, and the acetyl-CoA is ready to go into the next step. The other thing I have in this process are, again, 2 hydrogen taxis, 2 NADs. Each one picks up a hydrogen and carries those 2 hydrogens over to their destination, the electron transport chain. So what came in? My pyruvate and some coenzyme A that was in the matrix. They combine and they break off 2 carbon dioxides. Those are exhaled as waste. They break off 2 hydrogens that get picked up by the hydrogen taxis NAD, become NADH, get carried to the electron transport chain, and I'm left with 2 acetyl-CoAs. Those 2 acetyl-CoAs are going to go into the Krebs cycle. So in the prep reaction, I'm just preparing, I am making my pyruvate and turning it into an appropriate form that it can go into the Krebs cycle. So I'm not getting any ATP out of this. I'm getting carbon dioxide, some waste product, getting my hydrogens over to the electron transport chain, and the final product, acetyl-CoA, I have 2 molecules of that, 1 from each pyruvate, go into the Krebs cycle. So my Krebs cycle, again, it's a cycle, so my final product in the Krebs cycle becomes one of the initial products when a new input comes in, and then I go through the cycle again and end up with the same thing, and a new input comes in and I go through the cycle. So the steps in this cycle are my acetyl-CoA comes in and it joins with the final product of the last cycle. That final product is oxaloacetate. The oxaloacetate, when it joins the acetyl-CoA, becomes citric acid. And that's why this cycle is also called the citric acid cycle. Krebs is the name of the researcher who worked out the steps in this process. So it can be called the citric acid cycle, or the Krebs cycle, or the citrate cycle. So you may have realized that any words ending in ic, acid, can also be an ate. So citric acid, citrate. Pyruvic acid, pyruvate, synonyms. So the citric acid cycle or the citrate cycle, the inputs are my acetyl-CoA from the prep reaction. That joins with the final product of the cycle, oxaloacetate, to become citric acid. That's my first intermediate in the cycle, and that's where the cycle gets its name. And then I do a series of conversions, changing that citric acid to a whole bunch of other intermediate products. And along the way, I am going to break off carbons, oxygens, and hydrogens. So along the way, I'm going to break off 4 carbon dioxides. So there go 4 more carbons and 8 oxygens. I've already released 2 carbon dioxides in the prep reaction. So all my carbons are now broken off from my hydrogens. So I release 4 carbon dioxides. I have a whole bunch of taxis in here. Right? So I have a new taxi company, the FADH company, that's going to pick up hydrogens to become FADH2. And I have 2 of those. So take 2 of those hydrogens over to the electron transport chain through that company. And then 6 NADs will pick up hydrogens and carry them to the electron transport chain. So now all 12 of my hydrogens from my original glucose molecule are on the membrane. Right? They're on that cristae, ready for the electron transport chain. All my carbons from my original molecule have been exhaled out. They're gone. And And most of my oxygens, I still have 6 oxygen molecules that are unaccounted for that we'll use in just a minute. The electron transport chain, just like in photosynthesis, I am going to take all those hydrogens that I dropped off. I'm going to pull out their electrons and push the hydrogens to the other side of the membrane. I'm going to build a concentration gradient using the energy from those electrons that really want to get back with their hydrogens. So these membrane proteins are going to kind of play hot potato with the electrons to keep them away from their hydrogens. And the energy from that electron motion lets me build that concentration gradient. And when I'm finally done with it, I'm going to release the electron inside the matrix. And now I'm going to let all those hydrogens flow back across the membrane. But they can't just flow anywhere. They can only flow through my ATP synthase. So those hydrogen ions flow in, they reconnect with their electrons. They are happy now. But we don't want to have that high hydrogen concentration. That'll change our pH. We need to get rid of these hydrogens. And we're going to use those last 3 O2 molecules to combine with hydrogens to form water. Right? So that oxygen's role is the final electron acceptor. Remember, anytime you see the word electron in this process, we are talking about that hydrogen atom, because my hydrogen atom is 1 proton and 1 electron, and that's all. So the hydrogen ion that I'm talking about and the electron that I'm talking about are all from that one, from the hydrogens. So my hydrogen has now reunited, and it's going to join the oxygen. It's my final electron acceptor. It forms water that is exhaled as an end product. And through that process, as those hydrogens flow through the ATP synthase, I'm going to generate about 32 to 34 ATPs in that process. Add that to the 2 I generated in glycolysis and the 2 that I got from the Krebs cycle, from the Krebs cycle, and I end up with 36 to 38 ATPs. So those are, we cover all of these. The role of NAD and NADH, FADH and FADH2, those are my hydrogen taxis. Oxygen is the final electron acceptor. My ATP yield at each step. So that is aerobic cellular respiration. We looked at fermentation is the process. If there is no oxygen present, I can't get into the mitochondria, and that means the prep reaction, the citric acid cycle, and the electron transport chain can't happen. What would happen is those 2 NADs with 1 glucose molecule coming in, those NAD taxis would be full with their hydrogens. They wouldn't be able to go and drop them off. Once those are full, then every other glucose molecule that gets broken down would release hydrogens, and I would become really acidic, and it would kill the cell. So if I don't have oxygen, my bypass is those NADs, my taxis drop off the hydrogen on the pyruvate and form lactic acid, or they drop off the hydrogen on the pyruvate and a carbon dioxide is broken off and they form alcohol. So depending on the organism, animal muscles form lactic acid, use lactate fermentation if there's no oxygen present, and yeast and some bacteria do alcohol fermentation. And so that's what we looked at in lab. So fermentation, this is, hey, I still need energy, I'm still a cell, I still have metabolism, and right now I don't have any oxygen or enough oxygen to go through the whole process. This is my emergency kick in, still give me some ATPs so I don't die. Emergency method till oxygen is present. If oxygen is not present in aerobic organisms, these will eventually, organisms will eventually die. But this gives me a stopgap until hopefully oxygen's present again. Let's see, and then the final part of this chapter, Look at that figure. There's a figure that shows a pizza and all the different things. We always show the equation for glucose as being our starting product for cellular respiration. But those monomers, polymers, are all made of schnapps. They'll have carbon, hydrogen, oxygen present. So really, I can put any of those biomolecules into cellular respiration. They would just go in at different points, and they would require the addition of a lot more ATPs to get those molecules ready to go in. They'd go in at a later point, not the start. We'd have to build the enzymes needed. So we'd have to put in a lot of energy so we don't get as much energy out. We always show the equation with glucose because that is our preferred energy source. It's what we always are prepared to do on a cellular level, is to use that glucose, but we can use all those other molecules as well. Take a look at where each of those go into the process, but we can use all of them for energy. Glucose is our preferred happy energy molecule. Questions on photosynthesis? All right, our last chapter 9, this is pretty straightforward. I know in your lab it combines mitochondria, sorry, cell respiration, mitosis and meiosis, but I really strongly urge you, learn mitosis first. This is why we separated out chapter 9 into unit 3, and then chapter 10 into the next unit. If you know mitosis, it'll be a lot easier to learn meiosis and keep it separate and distinct from mitosis. So separate that lab out. So the cell cycle, this is how we go from 1 cell to 2. And the purpose of mitosis, our cell cycle, is to increase the number of cells we have. So that's how we grow from a single cell after fertilization to 35 trillion cells as adults. The purpose of mitosis is growth. And I want every cell in my body to be identical genetically. I'm not going to use them the same. I'm not going to use the DNA the same. I'm not going to activate them the same. But all of my cells have to be complete and have all my DNA. So in the cell cycle, mitosis makes an exact copy. Mitosis is cloning. So I'm going to make an exact copy of my cell. And then I'm going to have 2 cells, and then those 2 are going to become 4, and eventually you become an adult with 35 trillion cells in your body. So we want to know the 3 stages of interphase: G1 or growth 1, S, synthesis, growth 2. Different cells, how long cells spend in each stage is different depending on the cell. Some cells will stay in G1 forever. Our nervous system cells, our nerves, stay in G1 forever. They never divide. Once we have our full complement, they grow, they can get longer, they can grow new synapses, but I don't get new ones. So we call that G0. Skin cells, we are constantly replenishing. Mucous membrane skin cells, they're always dying and shedding off. So we're going through that stage pretty quickly. And other cells will spend longer periods of time in G1 because that's where they do their job. So that's where they're doing whatever their job is. All our cells have a function, and G1 is where they do that job. But when it's time for more cells, they're getting worn out, I need to replace them because they're old, they got damaged, I need to cut myself, I need new cells to replace those damaged cells, or I just need to grow. I'm going to say, oh, time to add more cells. So I'm going to go through the S phase synthesis, and I'm going to make a copy of my chromosomes. Each single chromosome can be referred to as a chromatid, and in the S phase, I make a copy of that. The exact copy, Xerox copy, stays stuck together to its original. I call those sister chromatids. And I want a Xerox copy so that I can share it with daughter cells after I divide. Right? So like a Xerox copy wouldn't double the number of pages of homework. If I give you 1 page of homework and I make a Xerox copy of it, you wouldn't say, oh, I've got twice as much homework. So you have the same amount of homework, but now you have an extra copy that you can share with a classmate. The S phase gives us a copy that I can share with a daughter cell. And then the final stage, G2 of the interphase, growth 2, I'm going to make all the proteins that I need and prepare for mitosis. Mitosis technically is nuclear division. So the steps of nuclear division begin with a nucleus and end with 2 nuclei. And then I go through cytokinesis to divide the rest of the cell components into 2 daughter cells. Checkpoints, figure 9.1 talks about checkpoints. This is pretty important that we have all the parts we need in our daughter cells. So the cell is going to stop periodically during the process to check and see if everything was done correctly. So after I've completed mitosis and cytokinesis and I have 2 daughter cells, I enter G1 and I stop for my first checkpoint and say, hey, did everything go as planned? Did everything divide out? I have 2 nuclei with all the chromosomes I need. Yep, check. Let's keep going. If not, the cell says, ooh, there was a mistake. And we don't need wonky cells with mistakes in them, so we're going to undergo apoptosis, programmed cell death. The cell will kill itself because there's an error. If everything was fine, it will continue on to the S phase. And at the end of the S phase, it's going to stop and do a check to see, hey, were all of the chromosomes copied correctly? Are all my sister chromatids all there? Yes, okay, we'll continue. No, apoptosis, because there was a mistake. Don't want mistakes in our cells. All right, so we went on. Everything's going good. We go through G2, enter mitosis. And in mitosis, we have another check during metaphase. Are all of my sister chromatids lined up in the center, and are they all connected to spindles, to my spindle fibers at either end to be pulled apart? Yes. All right, proceed. If not, boom, apoptosis. So our cells have built-in ways to protect our bodies and ourselves from errors, from cells that will be wonky or won't be able to function. Let's see, chromosomes. Those are the DNA and proteins that make up our genetic makeup, our genome, and chromatin is what we refer to them, to the uncondensed. So these are just like all spread out like yarn. DNA and proteins, and then chromosomes, we've organized them by wrapping them around those proteins to organize them like a nice skein of yarn. In eukaryotes, we talk about the diploid number of chromosomes. Di is 2. Diploid, I have my complete set of chromosomes. I got 1 set from my mom and 1 set from my dad. That makes me a diploid organism. 2N. In humans, the 2N number is 46. I get 23 chromosomes from mom and 23 from dad. Different organisms have different number of chromosomes. So we don't want to say 46 all the time because not all organisms have 46. So use 2N makes the discussion universal. Doesn't matter what organism I'm talking about. For eukaryotes, the complete complement of chromosomes is 2N. The haploid number, N, half, I think half. Half come from my mom, half come from my dad. In eukaryotes, the only haploid cells are going to be my gametes, eggs and sperm. And that's when we get into meiosis, we'll talk about that. But the haploid number, N, tells me how many chromosomes I got from my mom and how many chromosomes I got from my dad. Look at that image of the sister chromatids. So we always see that kind of X-shaped chromosome. That is my chromatid after the S phase when it's got a Xerox copy attached to it. And it attaches at the centromere. The kinetochores are the little handles that my spindle can attach to. The centrosome is my mitotic spindle organizing center. That's what's going to make my spindle fiber. These are those 2 barrel-shaped organelles that we learned about in chapter 4. Each barrel is a centriole. Together they're the centrosome. They replicate between the G1 and S phase so that they can So I have 2 in each cell. Then when my cell divides, I'm going to have a centrosome in each daughter cell, ready to go again. Plus, I'm going to have one at opposite sides so that spindles can form on either side and pull my chromosomes apart. There are 5 phases of mitosis. In my presentation for lab, I kind of combined prophase, prometaphase, because this is continuous. It's not a snapshot, prophase, prometaphase, metaphase. It's this continuous progression until I end up at the end of telophase with 2 nuclei in a cell. And then begin cytokinesis with that cleavage furrow forming. Early, my first phase, the chromosomes condense, the nuclear envelope breaks down, my centrosomes begin to form an aster. That's the original, those spindle fibers starting to come out from there, and they start migrating to opposite poles, the 2 centrosomes. Prometaphase, my centrosomes are close to opposite poles of the cell. The spindle fibers have reached out and connected to my chromosomes at the kinetochore, my sister chromatids. And then those spindle fibers push and pull until those sister chromatids are aligned on the metaphase plate. And that's metaphase. My sister chromatids are lined up in the middle of the cell on the metaphase plate, the dividing line. Then those spindles start to pull, and they pull apart the sister chromatids in anaphase, A apart. Telophase, those chromatids are at opposite ends of the pole. Nuclear envelopes reform around each set, and the cleavage furrow begins to form. And that continues to pinch in, kind of like a belt tightening, and my cells pull apart till I get 2 separate cells around each of those nuclei. I can't pull my cell apart in plants because of the cell wall. It's rigid, won't let that process happen. So I'm going to form a cell plate by making these vacuoles that have the precursor molecule for cellulose inside. They're going to line up. Their membranes are going to merge. It's going to divide me into 2 cells. Remember that membrane's the boundary. And the stuff that was inside those vesicles will form cellulose cell wall. So the functions of mitosis are for growth and repair and replacement of old cells. The last portion of this chapter talks about stem cells. Stem cells continuously conduct mitosis because they haven't differentiated yet. They don't know what they're going to be. So every cell in our body has all the directions for everything it could be. But as we progress in our development, the cells figure out what they are going to be when they're adults, and they turn off some of those genes. So some of the functions they have instructions for, they can no longer do once they've differentiated into what they're going to be. So if they differentiate into a nerve cell, they're going to turn off all of the genes, all of the instructions for anything other than conducting nerve impulses, making neurotransmitters, the things they have to do as nerve cells. Prior to differentiating, we call them stem cells because they could activate any of the genes possible. And that's why those are so important in research, because it's one of the only cells that could possibly become a new nerve cell, since our nerve cells never divide. You want to read more about that on page 157. Cancer is unregulated, uncontrolled mitosis. So if my cells never spend time in G1, never do their job, they just keep hurrying through that cycle to get to mitosis again, to divide, no regulation, no apoptosis. We call that cancer. And then talks about benign versus malignant growth. You want to read that portion on cancer. Tumors, just unregulated cell growth till I have a mass. Metastasis is one of those unregulated cells, breaks off, travels to somewhere else in the body. Angiogenesis, I become vascularized. So my tumor has blood vessels flowing through it, so I can get food into that so it can continue to grow and get wastes out so that those cells don't die, so they can keep going through this unregulated growth. And look at our proto-oncogenes, early genes developing cancer, tumor suppressor genes. And my telomeres are those end portions that get shorter as we get older. And then binary fission and asexual reproduction in prokaryotes. Very similar process to mitosis, but remember mitosis, the definition is nuclear division. We have no nucleus, so we don't actually have to separate out a nucleus. We just have to make a copy of our DNA, elongate the cell, separate those, and then divide the cell. All right, so I think between lab and lecture should be good on those. And we're a little bit over our time. Any questions on the cell cycle and mitosis? So mitosis, whatever number of chromosomes I start with, I end with the same number, but in 2 cells. The way I get the same number of chromosomes in 2 cells is by making an exact Xerox copy during the S phase. And then I separate out my Xerox copies. All right, well, good luck on the exam, everybody. And email me if you have any other questions. All right. Thank you guys for coming in and listening in. Exam 3 is open now, and it will be available until midnight on Sunday. And so we'll start our Unit 4 on Monday, and then we're starting it now because there's a lot to cover in our last 2 weeks of class. So, but we have some fun topics coming up, so a little bit easier. This really is one of the more challenging ones with cell respiration, photosynthesis. So, all right, so good luck. You guys have a good day too.